Hello. Today's objective is the following. You will be able to trace the general process where the progeny from a single cell forms an embryo in which the cells multiply to form the many specialized cells, tissues, and organs that comprise the final organism. And this is a standard, which is pretty comprehensive. We're going to complete that today using our PowerPoint notes. Okay, so first thing, we're going to put this light off, get a better view here. Okay, so the first question that we have, which really zeroes in on our standard, on our objective, is how does one cell become a complete multicellular human with trillions of cells? So you ask yourself that question. Well, first of all, it begins with the sperm and the egg cell. And what we have in this union is really the beginning of life. And it doesn't occur very often, 1 in a million chance, which you probably think that that happens very often considering the billions of people on Earth. But in reality, the chances are overwhelmingly against it happening if you consider the amount of sperm that's produced versus the egg that is ultimately fertilized. And so what we have is sort of like two half cells that become one whole cell. Even though that's not very accurate, a sperm cell by itself is a complete cell with 23 chromosomes, which we'll talk about later on. And an egg cell, it's a complete cell with its own number of chromosomes, which is 23 for human cells. So with that process, fertilization, our first cell is called a zygote. Okay, and so one sperm enters the ovum, which is another word for an egg. The ovum changes dramatically, preventing any sperm from entering it after the first one penetrates that cell membrane. The egg and the sperm unite, forming the zygote, and a zygote is the first complete cell of a new organism, in our case of a human being. See if I can get this over here. See if this works as well. Okay, there we go. And so after that zygote forms, cells begin to divide. And this process of division, where one cell becomes two and two becomes four, etc., is called mitosis. And we will also learn about mitosis later on. Not in this objective, but soon after. Chromosomes that were originally in that first cell, if your organism originally had 2 chromosomes, if you see the cell all the way on the left here, okay, after mitosis occurs, you have 2 new cells which are identical to the original cell. And identical means that they too will have 2 chromosomes in each cell. Okay? So if you think about the math, it sort of doesn't make sense. You say, how do you go from one cell that has 2 chromosomes to 2 cells that have 2 chromosomes? Well, the answer to that trick is in this process called replication, where those chromosomes, which really have what molecule in them? Let me give you a couple of seconds there. The molecule that makes up your chromosomes, that contain all the genetic information. Did you say DNA? Okay, Jeopardy music is over. Yes, the DNA material doubles itself, okay? And so one chromosome becomes a doubled one with double DNA. The other chromosome double itself. And then eventually when the division does occur, that's how the math adds up, where you start out with 2 chromosomes or 2 DNA molecules. They double up, you have 4. And now when the cells divide at the end, you have 2 molecules of DNA or 2 chromosomes in the resulting cells. Okay? And these are the progeny cells, cells that result from the original cell. Okay. this process from division, the process occurs in a way that cells start to multiply, and depending on the number of cells, we have different stages that has names. For example, day 1 through day 4, those cells are in that combination is called a morula. Okay, and the number of cells doubles each time. You go from 1 to 2, etc., to 4 to 8 to 16, 13. And at that point where we call it a morula, it's because a solid ball of cells. And at this point, all the cells are the same. Okay, so you think about that for a while versus, you know, who you are today, where all your cells are very different depending on the specialization, okay, which we'll talk about in a few seconds. From day 5, that morula starts to change. It keeps on dividing, but now it starts to form a hollow area. And now, because of that hollow section where the cells start to push off from the inside out, forming this hollow section, we call this stage now a blastula. And if we continue, from day 6 to day 7, this is usually at the time where the fertilized egg starts to implant itself, around day 6 or day 7 in the uterus. Okay? And hormones are produced during this time which prevent the menstrual flow, allowing the female body to use that blood for its purpose at this point, which is to sustain the life of this developing fertilized egg, which now is called a blastula. By the time that you get to day 6, 7, you have implantation. And again, we'll continue with the next stage, which is gastrulation. Okay? During gastrulation, this hollow blastula folds in, starts to take yet another shape. Now it begins to have 3 distinct layers, and these layers are really the beginning of the differentiation process, because that ectoderm is the outer skin layer, okay? The mesoderm, which is going to be in the middle part of this diagram, becomes the middle skin, and the endoderm becomes what's also known as the inner skin. Okay? And so to summarize this segment of the video, from the zygote, we have the stages that will produce 8 cells, many more cells, which we now call blastula, which has a hollow ball, and before that the morula, which is not listed here. Okay, so the solid ball of cells, the morula. After that, the blastula that has the hollow center, which is called the blastocele. And eventually those cells start to move, and you have 3 distinct layers. Okay, the distinct layers are the ectoderm, the endoderm, and the mesoderm, which is not illustrated here. And then we get to the gastrulation part, where the fold becomes even more distinct. And we're going to stop here, and we will continue with the second video, with the second part of this lecture.